So this is the workshop number 8. And today's topic is looking into large proteins. And mostly I'll be highlighting techniques, which is often referred to as methyl-NMR techniques. So this will aid us in looking into very large proteins. So before we start, these are the BioNMR core workshop etiquettes. So this webinar is being recorded, and we will edit it and share it on our website later. So we can look at it later as a resource. Webcams are optional for hosts and participants, but I have my webcam on. If given opportunity to be on the stage, please watch your mic if it's mute or not. And please use the Q&A panel to ask questions, raise your hand. You can make it as interactive as possible. And feel free to raise your hand and talk to us if you're not able to understand something, or if I'm going too fast, or if it's something that needs a little more clarification, let us know. Okay. So this is the overview of today's talk. I have only 40 slides, so this could be over very soon, but that also means that I can go into any amount of detail if needed. So stop me if that is not clear on what topics I'm covering over here. So the first thing we'll look at is what the problem is. Are large proteins, is NMR easy for them? What are the few NMR strategies that we can use to analyze large proteins? Then we'll look into methyl-NMR, and we'll try to push the limits of NMR using the methyl-NMR technique. And then the big question is, how do we assign these signals that you see using these methyl-NMR? And can we then, once we assign, use the assigned values to get some idea on the dynamics of things that people are interested in, especially involving biomolecules? Okay. So the question here is, large proteins, is it easy to do NMR of them, right? So right on the right side of the slide is a huge protein. This is 1.2 megadalton protein that is made up of multiple domains. Some of them are homodimers and tetramers, and these form this ion channel, and they bind to much larger proteins. So those are the ones shown in red. And this is one of the systems worked by Louis Kay's lab. So this is a huge protein, but then the conventional NMR methods would be a problem for this. So when you work with such large proteins, you will have slow molecular tumbling, and you will have a fast NMR relaxation because of the size of the protein. And you will also have extremely large light broadening because there are so many protons around. And you will have like proton-proton relaxation happening. And also you will have much higher, much lower signal to noise compared to small proteins. And then you will have low resolution because there'll be way too many resonances to look at, and sometimes they tumble so slowly that they don't even are broadened out because of the slow tumbling, that you don't even see these signals. So only signals you will see are from the proximities of these proteins, probably the end regions that are kind of very flexible. Okay, so conventional NMR methods is a problem because of all these issues that you would be facing when you want to look at every proton that makes up this huge protein. Okay, so it's be extremely crowded, and it makes a problem of assigning resonances compounding as you increase the size of the protein that you're like trying to look at. Again, I'm just using protein as an example. Any large biomolecule will have a similar problem. Okay, the more protons you have, the more problems that you have. You'll have more resonances that you need to assign. So what are the few strategies that you can use in NMR to analyze these large proteins? Okay, so the first strategy is probably you want to go to a higher field. So in the slide, I'll show you how you can actually leverage the 1.2 gigahertz NMR that's in Ohio State that you can use. So all you see here on the left side of this slide is that as you increase the field strength, you're going to increase the T1. So that kind of helps a little bit when you want to look at large biomolecules. And also, as the rotational correlation time increases, you're going to have issues with the T1. The easy solution is go to a higher magnetic field, and NSF has funded the installation of this 1.2 gigahertz that is capable of doing both solid state as well as high resolution liquid state NMR in our gateway belt area, which is in Ohio State, totally funded by NSF and is open to all researchers to use who are in this area. So um, go ahead and look into what this resource is. The website is over there, and I can put a link later, and you can look at it. But the idea of going from, let's say, 600 or 800 megahertz to this higher field means that you'll have higher resolution, you have higher sensitivity as compared to a lower field magnet, as well as you will have better T1 values for large biomolecules. So you can analyze them using this cryoprobe. So the picture there that you see is actually the cryoprobe installed on a liquid state system. So basically, you can use that cryoprobe. Again, the software is no different than what you're used to on the 600, 800 that we have here at U of M. So you can use this particular instrument if need be, and if you are looking at very complicated problems as well as huge proteins that are not feasible using the current instrumentation that you have at U of M. Okay. The other technique that people have developed is something called transverse relaxation optimized spectroscopy, also known as TROSY spectroscopy. So basically, in TROSY, you in an HSQC experiment, you have four different components. So you have a fast relaxing component, and a slow relaxing component, and you have two other components that are somewhere intermediate. Okay. So when you run actually a HSQC experiment, you get a combination of all this. So you actually see all these components when you have no decoupling in both proton as well as nitrogen dimension in this case. So once you do the decoupling, everything collapses and you will get one peak, and each peak corresponds to the spin system HN in this case. And this could also be proton carbon spectrum as well. But the thing is, well, once you like optimize this experiment, you will get one signal. But this HSQC signal would be a combination of relaxation values of all these components. So basically, you'll have lower sensitivity because you also have the fast relaxing component that contributes to the signal. So in the TROSY experiment, what you can do is select only this particular component of the four components that are present. So we use something called phase cycling, and we can eliminate these guys out and we can combine them. So when you run a TROSY experiment, all these components cancel out and you only end up with one particular component that is the slowest relaxing component. Slower relaxation means stronger signal, and you get much higher signal to noise. Okay. Another thing to note is don't truncate the TROSY with less number of scans. Just go and look at what is the minimum number of scans required to complete the phase cycling, or else you will start seeing these components show up here as well if you don't complete the complete phase cycle. Okay, so come talk to me if you have issues. Once you run your TROSY, you see more than one peak for each amide, then we have a problem, and we should be able to easily rectify by looking if we have not completed a phase cycle. Okay, so this is one of the techniques that you can use. So all this means is that you're going to get stronger signal, and you're going to get the same amount of signals as the HSQC, but it'll be sharper. So you can kind of push the limit a little bit by using this particular technique. It doesn't take any extra time. except for making sure that you have the correct phase cycle. Usually it's 16, but then you need to be a multiple of that phase cycle. So that's the only thing. But if you have a choice between running a TROSY experiment and an HSQC experiment, I would choose TROSY experiment. The one thing to note is that this is the separation between these components is 1 over J of whatever you're looking at. So this is 1J HN. So that means that the TROSY component of every signal as compared to HSQC will be separated by half of 1 over J, the half J between the two the components that we're looking at. So make sure that you take that into account. That means all peaks are shifted a little bit, okay, both in your nitrogen as well as proton dimension. Again, the absolute position is not important. It's that you see signals from your protein. So basically, you can run TROSY versions of pretty much all the experiments that we have been using. So you can run TROSY HNCO, HNCACO, and all that. Okay, so we have a workshop on that that we have already done. So you can look into it, and you can run those experiments to do the assignments in your amide HSQC experiment, okay, which is converted now in amide TROSY experiment. Okay. So the next technique that you can actually help with is use something called band selective excitation shock transfer, also known as best NMR technique. So in a protein, you have your amide region, and then you have your side chains. Then you have a huge water peak that is in here. Usually proteins are in aqueous-based samples. So you'll have your water peak as well. So the idea behind running best experiments is you only excite the region of your interest. So that'll be this region on your spectrum. So you don't even excite the side chains or the water. So that means that you don't have to wait for the relaxation of water. You don't have to wait for the relaxation of the side chains to come back to Z to actually do the next scan. So this is kind of beneficial. And you can set the center of this particular best experiment as well as sweep width using the different parameters in your biotop, if that is what you use, or you can actually manually put in these values in your const 54 and const 55, which corresponds to the sweep width as well as center frequency for this best excitation. So that gives you the advantage of using much shorter relaxation delay. So in this case, instead of using 1 second relaxation delay, because you're only waiting for these amides that relax much, much, much quicker, you can actually recycle this much faster by using 1/5 the relaxation time from 1 second all the way to 0.2 seconds for getting the similar quality of your spectrum. Again, you can also combine this with the previous thing that I talked about. That is, you can do a TROSY with the best parameters in it. So we call it the best TROSY. So you can run that as well. So basically, you get the advantages of TROSY principle as well as you can look at you can do it much 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 faster for the same amount of time. You can have 5 times the number of scans, so you can actually increase the signal to noise by square root of 5. Okay, so that is the idea behind running these experiments. And because you're combining both best and TROSY, you can actually get push the limit of what you're looking at for the same amount of NMR time for collecting NMR spectra on large proteins. Okay. The other technique that people have been using is band selective optimized flip angle short transient experiments. Again, that's a mouthful, so we call that at so fast. And in NMR, we have these fancy names. So we call this so fast NMR. This also uses D1 values of somewhere around 0.2 to 0.3 seconds instead of 1 to 2 seconds. Again, the idea here is to actually do selective T1 relaxation, do Ernst angle optimization, and this is mainly used for highly deuterated as well as N15 labeled metal samples in water. And these experiments are from this particular paper from Kanadimo's lab, and they were gracious enough to give us these pulse programs, and we have them implemented on all of our spectrometers. All you have to do is type in so fast in topspin, for the experiment where you want to run for your sample. So basically, you put in your sample, do all the setup, that is, you lock, shim, tune, and get it ready, and then you type in so fast, and that brings up this particular window. So here it gives you a list of different experiments. So the let's say you just want to run an amide HNHNQC, you type in one as an input. So once you type that, and then it'll set everything up for you, and then you can just do RGA and ZG. Okay. So basically, these list of experiments not only have amide, it also has methyl, it also has experiments for assigning aromatic rings. And here is the guide for it. So basically, you also have a bunch of these experiments that talk about C-methyl, H-methyl, assignments for all that, and different experiments that you can use to do these assignments. Again, you can refer to this particular paper, fantastic paper that covers pretty much the wide variety of experiments that you can run to do your assignments. Again, I will talk about this in more detail in the few slides. But yeah, you have this suite of experiments on our spectrometer. Okay. So again, you can use this to actually see that using the conventional methods, you probably get spectrum that looks like this. Again, there's a more complicated 3D NMR for doing a nosy experiment to do some method assignments, but you can see the exact same amount of time, you can get much higher signal to noise as well as much better resolution because you get better intensity of these particular signals that you can owe to actually the so fast effect. In this case, in this paper, they were able to get a lot more NOEs. Using the conventional method, they were only able to get 800 NOEs, but that kind of like doubled when they were able to use exactly same amount of time to collect the so fast versions of the experiments they were running. Okay. So basically, this is another technique that you can use to actually get much better signal to noise and analyze larger proteins. Okay. So the next thing that you can also utilize is something called non-uniform sampling. Again, this is just a representative HNCO experiment. This is a 3D experiment wherein you correlate the proton, the nitrogen, the amide proton, and the amide nitrogen to the carbonyl carbon that is on the third dimension. Okay, so there's the carbonyl carbon, nitrogen, and proton, and you see these signals. And the one thing that you will note here is that the signals are kind of quite dispersed and sparse. So basically, that means that you can take this as an advantage. So that means that you don't have to sample every point in this cube to get a three-dimensional experiment. If it's a 4D experiment, you again don't have to sample everything. Instead, you can just sparsely sample these. So we have this mathematical way of actually collecting these spectra with only, let's say, in this case, 25% of the points. So here you see the distribution of points in one dimension versus the other dimension. These are the indirect dimension. And of course, if this is an proton detect experiment, so this is your direct dimension. So what we're talking about here is the points that you collect in your indirect dimension. Okay. So this is the distribution of the points that you collect. So when you like when you do the calculation of doing the Fourier transformation, you can use mathematics to actually figure out what the signal is going to be, and then recalculate these signals. And basically, you can then get a spectra that looks like this for spending only 25% of the time. Again, you can combine all this. So you can combine this NUS with the best and the TROSY, or you can combine NUS with your so fast HMQC and HSQC experiments. Okay, so and also three-dimensional and four-dimensional. The more dimensions that you have, you're going to push the limits of increasing. That is, you're going to utilize the time in a more better way because you're not collecting all the points, and the more dimensions you have, you kind of are helping yourself by spending less time on the spectrometer to get a lot more data. Okay, instead of collecting 100% of all the points. This can be easily done by going to the parameter type FN type in your top spin. So you can type in non-uniform sampling over there, and then go to the bottom of your AccuParse and type in the percentage of sparse sampling that you have. So I would say start with 50% first and see how it looks. And then you can go down depending on your concentration of sample as well as how many signals that you have in the region that you're looking at. So it's a thing that you have to optimize. But yeah, so start with 50% and then you can go lower if that kind of helps with what you're trying to look at. And the other thing that you can also do is you can also set these things in Biotop as well. So that means that you can type in the percentage NUS over here in Biotop, if that is how you set up your experiments, wherein it automatically calculates how many points it has to do, which just matches this number. And again, this is combined with your best TROSY experiment. So basically, you have your best parameters over here, and this is the TROSY experiment. So you combine all that, and basically, for the same amount of, like, probably a fraction of the time, you get much higher signal to noise. So that means that you can effectively, like, get very good signals for the same amount of time when you combine all these techniques that I talked about. Okay. Okay. What can we do further, right? So the next portion of this talk is going to be talking about Methyl-NMR and how we can push the limits. We are going to combine this with the previous things that I talked about and what it is, what is Methyl-NMR, and how we can actually utilize this. Okay. Um, again, here I want to make a case for methyl groups as a probe for NMR. So these are excellent probes for structural characterization for large proteins. Why? Because they're very well distributed throughout the protein structures. They are pretty much everywhere. They're in the hydrophobic coats, they're in your macromolecular interaction surface, and I will bring these two up every time, okay? This is like a very good feature of these methyl containing amino acids because they're everywhere. And the other good thing about methyls is they are non-exchangeable protons. So unlike amide protons, protons can exchange with the protons that are in your solution, especially water, and you have the solvent exchange that could also contribute to line broadening. And in the case of methyl, these are non-exchangeable protons, so they do not exchange with your solvent, which means that you can have your sample in 100% D2O, and that will give you still better signals than a sample where you have an amide proton that is your N15 labeled proton connected proteins. When you put that in D2O, those exchange, and then you slowly actually lose out all the signal that comes from these protons. Again, you can use that for an advantage for doing hydrogen deuterium exchange spectroscopy. Again, that is a different topic, but if you run long experiments over a function of time, you're going to lose those signals. Okay, so in that case, you can use these as probes, wherein you can have it at 100% D2O, and it's not going to exchange with your protons that are in your methyl groups. And another advantage is each methyl has three protons, so you get advantage of getting signals from three protons rather than one proton in an amide. Okay. Um, the other thing is these have unique chemical shift based on the type of methyl. What I mean by that is you can look at the figure on the right. This is from a review paper that we had. So in this case, they are colored differently. So you have isoleucines in a region in your proton carbon HMQC experiment. So every signal corresponds to one isoleucine over there in magenta, and then the red ones are methyl, so they are in a very unique region as well. Same thing about threonine. So that is these orange ones. The blue ones are valine, and the green ones are leucine. Okay, so each one of these residue lie in a different region. Again, when you do these three-dimensional LMR experiments that we have talked in workshop number 3 and 4, you will notice that these show up there as well. So these are the terminal regions of the assignments when you do those sidechain assignments. Okay, so if you go to those workshop and look at the sidechain assignments, that's what we are talking about. Okay, so this is just the region where they'll show up, and these are pretty much a very unique thing about these methyl regions, you can look up at BMRB website where are these guys distributed, and they're usually in these regions. Again, I colored them differently so that you can see where these residues lie. Okay, and I will talk about the technique on how you can actually do this as well. The other thing to note is these are very expensive to make. So again, I'll show you what are the materials that are required, and I have a protocol that I can share with you when you want to make such a sample. Again, it's not difficult, and people have done it at large scale. People have automated it. I'll show you some examples of how that is done. And basically, please note that it's going to be expensive. Okay, each one of this requires a metabolite that you need to specially order from different vendors. I can give you those catalog numbers as well as vendors and all that information. Contact me, and we should be able to figure that out. Okay, and there are a lot of review papers that talk about this as well. I can point you to those papers too. Okay. So these are the advantages and the disadvantages. Okay, the other disadvantage is that, as you can see, this dispersion in proton dimension is not all that great. So you might have some overlaps. Again, that is okay as long as you're able to label them in a specific manner. And then we will also talk about some issues for IL and V in a few slides. Okay, probably this slide. Okay, so on the left over here, you have this particular protein. There's a huge enzyme. It's an oxidoreductase enzyme. So all the black dots that you see here is every proton that is there in this particular protein. Okay. And when you want to analyze this particular protein, it's going to make it a lot more complicated because you have way too many protons over there. So now let's strip out all these protons and just look at methyl protons. So you only have 108. So this makes it much more simpler, right? And then over here, we can do something called as just label one particular methyl in case of valine, leucine, and isoleucine. So we call it ProS and particular. You can use certain metabolites wherein only one of the methyl is labeled, which means that you get only one signal instead of two signals for each and every one of these residues. So that kind of simplifies it and makes one signal coming from I, L, and V. So basically, you can then reduce this number from 108 to 70. And then another important thing to note for this particular protein, where it does the oxidoreductase reaction, it's actually in this region. Okay, so that means that you have a lot of probes, especially these methyl probes, that are in this region of interest. So this can report on the enzymatic reaction or the structure or the interaction that is happening for this particular protein. Another example on the right over here is again from Lewis Case group. And in this case, they have huge proteins that interact with each other. So you have REC2 domain, REC domain, and HCH domain all come together. And basically, they have their own metal spectrums that you can do separately. But the important thing here is that this kind of simplifies the thing that you're looking at. So basically, instead of looking at huge number of protons, it kind of reduces the number of signals that you're looking at and makes your analysis of these huge complexes possible because this is a more simplified problem. And the other thing to note here is very well distributed. Okay, all these probes are very well distributed, and that means that they can still give you information on the interactions between these because there will be chemical shift changes when they come together. These are just separate samples that are made, but then you can combine them and see how they are going to affect each other when they combine and interact with one another. Okay. Again, this is a overall view of these two examples that kind of show that this is a very powerful technique, and you can like simplify your problem, and then you have techniques wherein you can label only one of them, one of the methods, and make your problem much more simpler and give you a better answer. Especially, you can look at much larger systems. So the technique that I always tell people, hey, we are running a methyl trozy, okay? And people say, oh, how do I set this up? Okay, this is not all that different. So this is just a regular HMQC experiment, which we refer to as methyl trozy, okay? So here is a regular HSQC experiment. So you have your transfer from proton to carbon, then you label your carbon chemical shift, and then you push back to the proton, and then you do your direct detection of protons. So this is a 2D experiment, okay? So that is the experiment in red over here. So this is again, from Louis Kay's group, wherein they talk about how you can actually use an HMQC experiment. Okay, so in an HMQC experiment, again, you have the same component over here, wherein you transfer the magnetization from proton to carbon. So here you have a multiple quantum term. In an HMQC experiment, at this point, you'll have a single quantum term. So that is what HMQC stands for, and HSQC is heteronuclear single quantum coherence. Okay, so then you again do the same thing that you do over here. That is, you label the carbon. So that labels this dimension over here. And then you again transfer the magnetization back to proton, and you do your direct observation. Okay, so the scheme is exactly the same for HMQC and HSQC, but the magnetization that you're looking at is different. This is a multiple quantum magnetization that you are labeling and observing. And here is a single quantum term, and you cancel out all the multiple quantum terms in an HSQC experiment. In HMQC experiment, you cancel out all the single quantum terms over there. Okay, but the advantage over here is when you have like huge, like this is like 2 megadalton protein, you have a much larger tumbling time. Tau c is just the larger the number, the bigger the molecule is. Okay, so as you increase the tau c, you see the HMQC still gives you some amount of signal. So that basically that means that you can use this to your advantage and collect this blue spectrum, that is your HMQC, for your metal label samples to actually probe your proteins. Okay, so for the rest of the talk, this is the experiment that we use. So we call this as a methyl trozy, but it's basically an HMQC experiment. Setting this up is exactly the same. You just type in so fast and you just set it up. I'll show you how to do that. But it's not anything different. So anytime I say methyl trozy, this is what I'm looking at. Okay, this blue spectrum. Okay. Okay, so the next question is, how do I label these metals? So what we do is, again, this is the E. coli metabolic pathway for the biogenesis of different amino acids. So here we're only focusing on amino acids that have the methyl group, and this is the biochemical pathway. So that means that you can manipulate the starting ingredient for making these amino acids. So you can manipulate the pyruvate. This was initially done, but that caused a lot of scrambling. But now what we can do is we can give different metabolites at the beginning of these reaction. And again, I can share the protocol with you at what point of time that you would add these metabolites. So basically, once it reaches a particular OD, you add this, and then once, let's say, you add this alpha ketobutyrate, you're going to label only the threonines. Okay, you're not going to label anything else. So you can make each protein with like you can make one protein with just threonine labeled. So if you use this particular metabolite, you can then just label isoleucine. If you use this metabolite, you can label leucine. Again, the same thing. You can use either one of these two to label valine, methionine. You can use this particular metabolite, and then alanine you can label by using a pyruvate. Okay, and you can then combine these different starting materials so to either label just, let's say, I, L, and V, or you can then just that is, you just use a combination of, let's say, threonine, isoleucine. So you can do those kind of like combinations of different labeled metabolites. Again, this is where it is expensive. You need to order these particular metabolites from a particular vendor, CIL, Sigma Aldrich, and a bunch of other vendors that you can use to label them. Okay, and you have to strategically decide which one you want to start with and how you're going to do this. Again, this is a more simplified thing. Instead of looking at that particular table, this is what you start with. Let's say you do alpha ketobutyrate, you're going to get your isoleucine. If you start with this particular metabolite, you're going to then end up with valine and isoleucine. So this just labels V and I. Again, for alanine, you can use this particular metabolite. For L-methionine, you can use that to label methionine, L-threonine to label your threonines, and these to actually do your Pro-S and Pro-R samples. So what this just means is that you just label one of your leucine and isoleucine, okay? So basically, because there are two methyls, you can just label one of them instead of labeling both of them. So that kind of simplifies the problem. Okay, so that is the one that I showed you before. Again, this is very well summarized in this particular review. You can go and read this. This is a fantastic review, but it kind of summarizes what I'm talking about. Okay. So let's say you make a combination. So this particular spectrum is just labeling threonine as well as isoleucine. So you get this. This is using everything. That is, you use all the metabolites to label it. You get this particular spectrum. You can just label valine and leucine. And this is a 30 kilodalton protein that we were looking at. Again, this 30 is again a small number, but this particular protein doesn't stay as a monomer. It stays as a dimer, okay, homodimer. So in NMR, you don't see two different signals corresponding to two different domains. That is a homodimer. You only see one set of signal. That means that they have the same structure, but they form this homodimer, which we have confirmed using other biophysical techniques. But then here, we look at this particular enzyme's reaction, and every particular methyl is labeled over here, and the color code corresponds to the different residue type. As you can see, the active site is over here, and this is an allosteric site over here, and they talk to each other, and we were able to analyze the allosterity by using methyl as a probe. And using conventional NMR method, this couldn't be possible, right? Because this would be like 60 kilodalton and very complicated spectrum when you want to look at every amide that is in there. Okay. Um, so again, I wanted to highlight that you can simplify your problem. You can create this Pro-S and Pro-R labeling for isoleucine, valine, and leucine. So that kind of simplifies the problem, that it reduces the number of, that is, every two peaks that you have, you can reduce that to one for these particular methyl containing amino acids. Okay, so now that you have labeled it, how do we assign these resonances, right? So yeah, so 2 slides earlier, I showed you all this, but then how do you know which signal over here corresponds to here, right? So we'll have some strategies that we have to look into to do these kind of assignments. So I'll stop here. If there are any questions, let us know. If not, we'll proceed. Okay. Okay, so let's proceed. Don't hesitate. Just start typing if you have any questions. Okay. Okay, so let's proceed. And okay, so how do we assign this? So backbone. So the first assumption is, let's say you already have your backbone assignment. So you'll be like, Deb, I already assigned this. Can I utilize that knowledge to actually assign the methyl group? So basically, you just have this labeled, right? So this particular thing is labeled, and you have this answer. That is, this is a known value. That is, you have an assignment of your proton as well as N15. Again, in some cases that is useful. Some people would have already had an assignment, so this is probably in the borderline. So maybe it is a 30 kilodalton protein or somewhere in that range where you have these assignments. So basically, this is a known answer. In this case, let's assume that you know this. So this is a proton nitrogen HSQC experiment, wherein you know that this is an isoleucine 240, right? So you run the combination of these two experiments. So basically, you would label the you start from the proton, go to this carbon, then move all the way down the side chain, then go to nitrogen, go back to proton. So you label everything. So there's a 3D experiment. So basically, you label this guy with your HSQC, then you label this guy with the HSQC. So these are the combination of those two. So basically, you can then connect those two to here. So that what this means is you have somehow encoded the carbon chemical shift in this particular spectrum and the proton chemical shift in this particular spectrum that corresponds to your proton carbon HMQC experiment. Okay, so basically, if you collect if you make this particular methyl sample and you want to assign this and you know this as an answer, you run these two experiments and you can connect them through this particular experiment wherein you label these two carbon and your proton. Okay. Again, this is only when you know your backbone assignments. It's good to have this information, but yes, this is good to use it. Again, the other way to do this is when you know your carbonyl carbon, C alpha and C beta. Look into workshop number 3. It's on our website. We talk about TROSY-based experiments on how to do these kind of assignments. So if you have those assignments, of course, you can also run a CCCONH in that case, or you can run an HCCH toxy, wherein you can connect your methyl groups to these C alpha and C beta chemical shifts. Okay. Again, this is the other technique that you can use. You can also run something called as 3D HMCM. Then you go down the chain, then you go to CB and CA. So you label that. So that is, let's say you have the chemical shift for CB and CA, or you have the chemical shift for carbonyl carbon. You can connect it back. Again, this is the paper which does this particular thing. Again, this is based on Louis Hayes' work where they have developed these particular experiments. These are available. Come talk to me if you want to set this up. So here in this case, you already have the chemical shift labels of C alpha, C beta, and carbonyl carbon that you can connect back to your methyl groups. Okay. Again, these two experiments that I talked about are called out and back experiments. You start from here, you go out, and you come back, and you label things along the way. And you can then use that to actually to your advantage to assign your methyl groups. Okay, so this is the other thing that people have been using. So these are a combination of two different 3D experiments. Again, this is not all that difficult to understand because, as you can see, this particular part of this experiment is an HMQC. So we call this HMQC nosy HMQC. So this portion again is a nosy, and this portion is an HMQC. So it's just combining a bunch of these experiments together and labeling things. In this case, you label the carbon over here. You then do your nosy mixing time. Then again, you label the carbon over here, and you do your direct labeling. So in this case, this is where it starts. So let's say it starts from this amide, then it transfers to this carbon. You label this carbon, bring it back, and then you do your nosy mixing. So any methyl that is close to another methyl over here, you're going to transfer that particular magnetization to that proton. From that proton, you go back to this carbon, label this carbon, come back and label it. So this means that for every peak that you have in your HMQC, you're going to have a carbon that is close by. So this kind of gives you a three-dimensional spectrum wherein you can actually see the self peak. So this is the plane for the resonance 1. So you're going to see itself. So that is this carbon over here. And then you're also going to see the carbon that is close by. So this is residue number 2. And you can confirm it the other way. So if you go to the plane of residue number 2, you will see the residue number. So this is the self peak, and you'll see that it is close to residue number 1. So this is how you can do these kind of assignments. So you can know that this particular signal is close to this particular signal in HMQC. And you can run a different kind of experiment. So we call this as the nosy HMQC. So in this case, you would label this particular proton, and you would do your coherence transfer. You do this carbon, bring it back to proton. So that's what you're doing over here. And then you do your nosy experiment. So you go from your proton to proton. And then this portion is just an HMQC, exactly same as here, or the HMQC that I showed earlier. So in this case, you would label So here you were labeling the carbon, here you're labeling the proton, and you can again combine this. This is again complementary. So you would again see the proton connections between the metals. Okay. So when you run these two experiments, it takes around 3 days each. Again, you can combine it with your NUS when you have large quantity of your sample, and then you can use that for your assignment. So this will kind of simplify a lot of things. The more dimensions that you add, it makes your life easier. And then you can also use NUS because you have more dimensions and make use of the NUS principle. Okay. Not only that, you can combine these two techniques. So here you are just labeling either carbon or proton. So you can combine that. So basically, this is a combination of that. So we call this HMQC, nosy HMQC. This is again developed, I think, at Bax's lab. Again, all those references are in this review paper, wherein you can do the whole assignment in one shot. So instead of collecting two different spectrums, wherein you just first assign the carbon, then you go back here and you assign the proton for your metals, here you can do that in one shot. All you can do is, oh, this is my peak in the residue number one, the self peak. It next connects to every other peak that is close to each other. So again, you can confirm it the other way around as well. Here is an example. Okay, so in this case, for this particular protein, you have your isoleucine 103. I will talk about how we assigned it. We use something called site-directed mutagenesis. I'll talk about what that is and confirm that this is this particular methyl containing isoleucine. And then we look at every other residue that is close by. So it is close to valine 9. It's not close to threonine 8, but it's close to methionine 1. It's close to leucine 10, leucine 104. Okay. So when you collect this particular spectra, you see that you can actually see every one of these peaks show up in the spectrum. So within one shot, you kind of looked at 103 and now you know what is close to it. And you can easily assign each and every one of these residue, and you can know that, oh, this is my valine 9, this is my methionine that is close to me. And that way you can utilize the crystal structure if you have it, or the cryo-EM structure if you have it, to again come back here and do the assignments. Okay. And then once we look at the perspective of valine 9, so we go to that particular plane. So valine 9, you will see that it is close to 103, right? So that shows up. Valine 9 is also close to threonine 8. So that shows up. That didn't show up here because that is kind of far from 103, but then you see that this valine 9 is close to here, as well as methionine 1. So basically, this is the assignment that can be easily done just by utilizing this particular one experiment rather than running two different experiments that is shown here. Okay, so it's just combining all that. Again, this is a 4D experiment, so it'll take probably a lot of time to if you do full sampling. So instead, what I would suggest is I can help you with setting this up, but then we need to utilize NUS. So that means that we need to have high concentration of your methyl label stuff, and you can run this one beautiful experiment, and you should be able to do your assignments very, very quickly. Uh, Again, this is just highlighting a protein. This can be anything that has methyls in it. So it can be your DNA, RNA, whatever the molecule of your interest. Okay. The the thing that I told you, that isoleucine thing, right? So how did I assign that? That is actually very simple. So we use something called site-directed mutagenesis. You get this as a kit. Here they kind of like went they use a lot of automation to make a lot of site-directed mutagenesis. There's one way to do it. You can utilize NMR techniques, or you can go and start mutating this out. Okay, so here what the goal is actually not to complicate the lives of a person who's making thousands of samples, but rather they were trying to look at what are good substitutions. So basically, if you want to assign that particular isoleucine, what is a good amino acid to convert it to? So basically, they give you good recommendations of converting an isoleucine to leucine. So basically, you will lose one of those signals in your isoleucine once you do that particular conversion. So in this case, you have two samples. One is your wild type. The other one is this isoleucine 162 that has got converted to leucine. So you lose a signal from isoleucine 162 once you have this particular mutation, but then you gain another signal when you have it as a leucine label. Okay, So once you do this particular mutation, you can easily guess, oh, this is the only thing that I changed, and the metal vanishes. So then you can say, okay, this particular signal is coming from this particular residue, and you go do this for various amino acids. So they do this for bunch of amino acids. So you would do this combination of different site-directed mutagenesis for each one of them. So each one is a prep, and each one is going to cost you a lot of money because you're going to use the precursors for that. And then the only thing that you need to do is just one sample, right? So you can get away by using a Shigami tube. We can make it highly concentrated, and you can make different mutations, and you can do the assignments. Usually, this is a good idea when you want to start doing your assignments. So this could be giving you those pivotal residues that you can look at. You can just mutate out the things that are in the center of a lot of things. So that way you can know for sure that this particular residue, by doing this mutation, corresponds to this peak. Okay, so you do this for strategically, and then combine it with this particular experiment. So basically, I know this is 103. So from that, I can, guess what the other residues are close by and do this assignment. So you can combine the set-directed mutagenesis with this 4D experiment. Okay. So not only that, you can then use this so fast experiment that I talked about. So you type it so fast and gives you every one of these 3D experiment over here for using the same technique instead of doing the regular HMQC, nosy HMQC. You do the exact same thing using the so fast principle. So that means that you can reduce the amount of time that you collect it, and then you can collect this as NUS as well, wherein you can just go in and put in the NUS values over there. And then using combination of those two, you can then push the limits and get these kind of assignments for your nosy metal. Okay, so these are actually showing you your metal assignments over here on the right. Okay. Uh, and the more signals that you have, the more NOE connections you have, you can get a lot of constraints that can give you information of what is close to what, and that's how you can do your assignments. Exactly the same way that we do for the your backbone assignments, wherein you use the nosy spectrum. Here are the different softwares that you can use. The Sina, there's a component over there that you can use. We have a license for this, but yes, it costs around 300 euros to get it. Here is a reference, and you can use a combination of ILVA to do this. But then there are different techniques. So I have personally used the Magma software. Again, it's free for academic use, and it supports a lot of different variants. So you can have it labeled as isoleucine, valine, leucine, methionine, alanine, and threonine. So combination of pretty much everything. And then using this, we were able to do the assignments for huge proteins. Okay. Again, these are all the new things that came after when we used this Magma. So these are newer techniques, and the latest one is probably this one, the mouse one. We will go over this, how this works. Again, this is free to use. They have a web server. Just upload your results over there and put in your PDB structure that you have obtained from crystallography or cryo-EM. So you pretty much know what the structure looks like. But then you can use that PDB to inform on where the metals will stay, and we can use that to actually do the assignments. I'll talk about that in the next slide. Okay. Again, all this is again reviewed in this particular review that I talked about, wherein they give you better some protocols as well on how to make your samples and also how to analyze your data. Okay. So most of them is free except for Sina, but we also have Sina license at U of M. So come talk to me if you want to use this. Okay. Um, Okay, so how do we do the assignments? We actually use something called graphs. Okay, so this is kind of, let's say, data graph. So you get a combination of, okay, so there's a leucine right next to a valine, that is right next to isoleucine, and this valine is close to threonine, which is close to methionine. So it's kind of like some kind of graph. Okay, again, this is taken with a grain of salt because you may not know the assignments, but again, some of them you might know because you would have done your site-directed mutagenesis. Okay, and then you go to the structure. So you do your PDB, you download the structure. This is that homodimer structure that I talked about. But again, from here, you know what is going to be close to what because this is the ground truth, right? So you know that leucine 10 is going to be close to valine 9, and that is going to be close to this isoleucine 103, methionine 1, and threonine 8. And then you will see that these two graphs look similar, right? So you combine those two together and you create this fitted result. So basically, you will eliminate out all the possibilities from here, and then take this assignment from the structure and put it in this graph. So you combine these two and you get your fitted result, and at the end you get your assignments. Okay, so this is how Magma works. Mouse does pretty much the same thing. This looks way too complicated, but this is in different sections. Okay, so again, this is very similar to this nosy thing, right? So basically, from your nosy, you construct this. Again, this is shown over here. So you can do a combination of different experiments wherein you get information on what signal is close to what. Okay, so you create a graph based on that. Then you pretty much do the same thing. You take the PDB structure. So all this that is shown over here. Okay, in B. So you take the PDB structure. From that, you create a combination of different conformation of these side chains, right? So you get a bunch of graphs. So basically, they will have these graphs, and then you combine this graph with these graphs over here that you got from your nosy. You iterate over and keep on doing that until you get a good fit, and it gives you gives you an output. So I think this Magma that I used was like a MATLAB component. So it's kind of cumbersome to get it set up. And if you don't have a MATLAB license, it's again a problem. Um, but I don't know what the particular version right now looks like. But the mouse one has a web server now, so you can just upload your results, upload your PDB, and it does this in the background and gives you the result. Okay, and it gives you a list of what your assignments are. So it makes your life much easier. You can go back and forth, and you can resubmit things. Well, if you're less confident or more confident on the NOE assignments that you do. Okay. But yeah, so once you do that, you can easily put it into this particular thing, and it'll do everything in the background and give you the result. Okay. So this is how you would use these software. Again, there are other softwares in the previous slide as well. So whichever you're comfortable with, you can use. But Magma and Mouse are kind of what I have used, and they're pretty easy to use as of now. What about dynamics? Okay, all we did now was just did the assignments, right? What will I do with these assignments? Can we use these assignments to look at dynamics of different proteins and stuff like that? And yes, we can. Okay, so we'll quickly go over different dynamic stuff over here. So basically, proteins are involved in huge range of interactions, macromolecular interaction, as well as vibration, as well as different things, right? And huge proteins lie in this range over here, and we can actually use these methyl assignments to get these kind of interactions. So the first thing that I would talk about is something called ZV exchange spectroscopy, which gives you information on second to millisecond time scale. So you have two different populations, PA and PB, and if there are things that are in equilibrium, the system goes from population A to population B and population B to population A, and you would collect this particular experiment. It's called a CZ exchange experiment, and you can fit the magnetization to get your conformation dynamics, that is the equilibration dynamics between these two states. Again, these are metal Trozy experiments that you use to collect this. Okay, you can use again metal probes to collect CEST experiments. Again, this is also in the similar range, seconds to milliseconds. So basically, there is a ground state and excited state, and there's an equilibrium between it. And this excited state is very lowly populated, could be even as low as 0.1 to 1%. And you can actually run the CEST experiment, bunch of 2D experiments collected at different points for your test values, and then you will fit that so that it'll actually a bunch of signals that you fit, and you will see that you can actually fit this to get your population of your ground state and excited state. And this is again developed in Lewis Case group, wherein here is an example wherein they show differences in the minor state and major state for different regions of the protein. Okay, you can use CES to methyl CES to do these kind of experiment. The other experiment that you would probably see a lot is CPMG experiment. This reports on a different time scale, so basically millisecond to microsecond time scale. Setting up the CPMG is very similar to your nitrogen CPMG experiment that you usually do. Again, I can help you setting this up, but here this is a methyl label sample. This is what it looks like. So again, this is an example from Lewis Case group, wherein this is it's NCP that is wrapped with a very stable DNA, and it's again wrapped with a different sample that is wrapped with rRNA. And you can see that they are slightly different, and you can actually measure your relaxation properties. The CPMG experiment can report on these two different populations because they experience different states. And you can use this to get your R2 effective. That gives you information on how dynamic something is. Okay. Again, this dynamics lies in this particular range, milliseconds to microsecond time scale. You can run something called zero quantum and double quantum relaxation experiments. Again, this is an example over here, a group in Japan where they showed how to actually label them with methyl for a huge protein, and basically use that as a relaxation, a fitter to a relaxation. So it looks pretty much like a T1 or T2 relaxation. Okay, so it's a bunch of 2D experiments collected for different relaxation time that you can fit to get your relaxation values. So you get this REX, and that can be indicative of different motions happening in this microsecond time scale. Okay. The last thing in this bunch of experiments is a double quantum, a triple quantum term, which is in forbidden state, and you have a single quantum term that's in an allowable state. You can allow the exchange between that, and that can give you information on the nanosecond to picosecond time scale. You can also do something called as deuterium relaxation. So basically, you have the CHD2 or CH2D components that you can look for different relaxation properties to actually look at the nanoseconds to picosecond time scale. Okay, so come talk to me if you want to run these experiments. These are looking at very high motions. Okay, motions in a much faster time scale. And last but not the least, you can also use these methyl experiments to do your PFG diffusion experiments. All this means is you start for magnetization over here, and you put a gradient, and you see if it moves farther away. The bigger the molecule, it tumbles slowly, and then you can fit that to actually get your diffusion rate constant. This is used for this can be used for much larger proteins. Again, if it is all protons that are labeled, you're going to have a lot of issues. So here you just label the methyls, and you can use that as a probe to do your pulse field gradient experiments. Millie has helped a lot of people run this for small molecules, but again, if you have these labels as metals, you can use that as a probe to do your PFC experiments for looking at large molecules. Are they tumbling as a monodimer, dimer, homodimer, all that kind of stuff? Okay. So come talk to me if you want to run these kind of experiments.